participant to come. Yeah. Okay.
Satria yang namanya usianya berapa Ibu? Usianya 28 bulan maka.
Oh yeah, please mute. Please mute your voice. Yeah. Rizky, we begins now.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, all participants, and today is the first day of online lecture series of Global Academic Program, collaboration from the Doctoral Program in Biomedical Science, Faculty of Medicine, Universitas Indonesia, SEAMEO RECFON, and the Rowett Institute, University of Aberdeen. Today we will be hearing a lecture from Professor Asmarinah, Professor Dr. Rernat Putranda Asmarinah, Prof Science. She is a lecturer at Department of Medical Biology, Faculty of Medicine, Universitas Indonesia, Research Coordinator at Department of Medical Biology, Faculty of Medicine, Universitas Indonesia, and the head of the study program of Doctoral Program in Biomedical Science, Faculty of Medicine, Universitas Indonesia. She is also has a lot of awards and grants from the Directorate of Education, and also she is a professor in biology in Faculty of Medicine, Universitas Indonesia. Today, we will be, the topic of today's lecture is the human genome. Okay. So all participants, please mute your microphone throughout the session. And this session is recorded and being live streamed to Doctoral Program in Biomedical Science YouTube channel. And also, if for the certificate will be sent after the participants joining all of the topics in the schedule. Thank you, and we will be continuing to our next agenda, which is the topic presentation from Professor Asmarinah. Prof, please, the time is yours.
Okay, yeah, thank you, Rizky, for the introduction. So, Assalamualaikum warahmatullahi wabarakatuh. Good morning for all of you, the participant from our program, namely Global Academic Program on Nutrigenetic and Nutrigenomic. The Master of Ceremony has already told that this program is a collaboration program between doctoral program in Biomedical Sciences, Faculty of Medicine, Universitas Indonesia, SEAMEO RECFON, and Aberdeen University. So actually this program is a lecture series for participant or student that can also take credit earning or credit for the education. Yeah, but some lectures will like today is open sit-in, free open sit-in for all participants that interested in this field. But the participant that interested to get the information or knowledge deeply about this field, nutrigenomic and nutrigenetic, yeah, you can enroll in our program for credit earning. This credit earning program composed or contain 4 credit point with 4 learning objective. At the end of my presentation today, I will explain you or I will inform you about our program for the global academic program with the credit earning. Yeah, for the first lecture series, yeah, I would like to inform you or give some information about the human genome, the basic knowledge to understand how the genome and or the gene interact with the nutrient or nutrition status, and can be influenced, yeah, the human health. So for the free sit-in program like today, we design or we give the lecture in the 5 topics, yeah, and this program will be held until December or until November, yeah, November. But for the student that take the credit earning, we have already arranged or designed the curriculum or the syllabus for this until 17 December. So this is just information for you all. And then, yeah, for 5 topics lecture series that is open sit-in. Yeah, begin with my lectures about the human genome. So, yeah, as head of the study program, a doctoral program in biomedical sciences, yeah, we would like to welcome all of you, the participants. Thank you for your attendance here. You're interested to enroll, yeah, in this seminar or this lecture, yeah. I heard that there are more than 200 participants that already enrolled in this program. I'm very happy. We are very happy, and we hope that this some knowledge about the genome and the nutrient and then its interaction can make or can give us better understanding about the interaction, this both field here, genetic and nutrition or nutrient, to give some understanding about the cause of the diseases, yeah, okay, in human being, yeah, of course. Okay, thank you again for your coming, your attendance here in our lecture, the first hour lecture in global academic program on nutrigenetic and nutrigenomic. So I begin with my slide presentation. And for student that take credit earning, there is 3 students in our program, study program. Please see my PPT or my lecture in the OCI, yeah, the special system information in our faculty to get some information and also the schedule of our lecture series. Okay, yeah. Can you see my slide? Okay, yeah, about the human genome. Yeah, this is yeah, a little bit famous term, yeah, because yeah, we our cell composed by genome, yeah. And then in this lecture, I would like to give some information deeply about the human genome and how the genome can be play a role, yeah, in the protein function, and also can influence also the human health. Okay, so the outline of my lectures, I would like to explain about what is the genome, what is the human genome, and then what is the DNA, gene and protein, and also a little bit about the human genetic variability and epigenetic. So I see or I read that the participant in this lecture is not only from the molecular biologists, but also from the nutritionists, that has maybe a little bit knowledge about the genome and the gene. Therefore, I would like explains also from basics knowledge about the genome, what is the DNA, what is the gene, so that the nutritionist that who has no information or knowledge about the gene can understand. I hope that all of you understand from my lectures today. So what is the genome? Genome is all genetic information of an organism that consists of nucleotide sequence or DNA, deoxynucleic acid. But some viruses like SARS-CoV-2 virus that cause the COVID-19, have a RNA sequence. Virus has two type of the genetic material, DNA and also RNA. And then SARS-CoV-2 virus has a RNA plus sequence. So as there is also a viruses that has DNA as a genetic material. So okay, the nucleotide contains the genome contains coding region that called as a gene. So and then non-coding region and this coding region or the gene and non-coding region located in the nucleus of the cell, and then in the natural condition, we call as this DNA sequence or DNA fiber as a chromatin. So and then the chromatin will condense in the mitotic phase to become the chromosome. So later I would explain also about the chromosome and then how many chromosomes that we have. So and then another genetic material or nucleotide in our genome, in human genome, is mitochondrial DNA. In the plant, the mitochondrial DNA located DNA, not the DNA, non-nuclear DNA located in the chloroplast, not the mitochondria. So some students sometimes forget it about the another nucleotide in our genome, because they forget about the another DNA material, genetic material located in the mitochondria. This mitochondrial DNA play a role also in some diseases if the DNA mutated. So now, we proceed to the what is the nucleotide? Nucleotide composed by sugar. There are three substances or three components from the nucleotide. Nucleotide composed by sugar. In the kind of the sugar is ribose that loose of the one atom of the oxygen, so named as deoxyribose. And then the second substance is phosphate group, and then the third is nitrogen base. To build the nucleotide, the nitrogen base is composed one of the fourth kind of the nitrogen base. And here is the four kind of the nitrogen base is adenine, thymine, guanine, and cytosine. And here you can see the nitrogen base. Nitrogen base actually divided into two kinds, purines and pyrimidines. And then purine also has two kinds, adenine with A letter and then guanine with G letter. So in nitrogen base kind pyrimidines. There are also two kind, yeah, thymine with the T letter and then cytosine with C letter. So this is the backbone or the structural, schematic structural of the nucleotide. Yeah, here you can see the deoxyribose, yeah, with the one oxygen loose, yeah, and then here is the phosphate group, and then here is the base, nitrogen base, yeah, one of this nitrogen base attached in this C carbon atom, and then so next with how the structure of the DNA. So once again, here is the composition of the DNA. Yeah, sugar and phosphate here, and then plus nitrogen base here is with the G or guanine. So this is the nucleotide. The unit of the DNA, unit of our genome is the nucleotide. And the nucleotide will be linked together, yeah, with the phosphodiester bond here, and then make a sequence, yeah, with the 5 accent to 3 accents way, yeah. So, and then if we see the normal DNA strand, actually the DNA is double strand, yeah, with the anti-parallel direction of this sequence, yeah. So here you can see, yeah, double stranded, and then here you can see there are 3 hydrogen bond between cytosine and guanine, and also there are 2 hydrogen bond between adenine and thymine. And naturally, the double-stranded DNA in the form of the double helix, yeah, with helix, yeah. Here you can see the double helix DNA structure. So there are also alternative form of the DNA double helix. Here you can see there are B form of the DNA, A the form of DNA, and also Z form of DNA. Here you can see the characteristic of the each of form, yeah, eDNA oriented like right-handed, bDNA also right-handed, but zDNA is left-handed. And there is also a difference between major groove and minor groove. What is the major groove and the minor groove? If I back to the last slide, here is the major groove. You can see here so big groove, yeah, in this double helix structure. But here you can see the minor groove. So major groove from the A form of DNA is deep and narrow, and then B DNA, moderate depth and wide, and Z DNA, very shallow, yeah, virtually non-existent, sometimes called a single groove. So like here, yeah, we cannot see clearly the groove, yeah, major groove in Z DNA. And then minor groove, here you can see also its characteristic of the form of this DNA. And then there is also some characteristic about the how many base in a turn, in one turn of this helix. Yeah, for the aDNA, there are 11 bases in one turn. Yeah, and then bDNA 10, yeah, about 10 bases, and then for the zDNA is 12 bases. So how can the DNA double helix with these differences of the form? Yeah, because the condition, yeah, because the different condition. So in the low humidity, yeah, with high salt, yeah, the DNA, yeah, the normal or natural form of the DNA is bDNA, yeah, with the low humidity and high salt. But with the higher, yeah, more higher the humidity, higher humidity until 95%, yeah. the structure of DNA will be to become the A form, yeah, with the low salt also. And then zDNA, yeah, the condition for DNA to form this DNA is if the condition with high magnesium chloride, yeah, more than 3 molar, NaCl or ethanol in presence of methylated cytosine and high humidity and low salt also. So this is just information for you. And then how the DNA packaging, yeah, package in the chromosome. Yeah, I have already told you that the human has 46 chromosomes, but how the DNA as genetic material packs, yeah, into the chromosome. So here you can see the DNA actually wrap the histone protein. So like this picture. So here you can see the four kind of the histone protein. It means there are 8 histone protein, yeah, wrapped by two round of the DNA sequence, yeah, wrap. And then there is also a special histone here, H1, to clock, yeah, this ikatan ini, yeah, the binding or the wrapping of this DNA to the histone. This nucleosome, yeah, this one nucleosome, the nucleosome, and then to pack together, bind together, become the chromatin, yeah, and chromatin, as I mentioned before, will condense in mitotic phase of the cytosol to become the chromosome. So, and then here, maybe with this picture, you can understand better how the DNA packs in the chromosome. Here you can see, if we see from the top, this is DNA strand, and then this DNA wrapped 2 times the histone protein, 8 histone protein, and then linked together this nucleosome. Yeah, this is one nucleosome linked together. And then this chromosome close together, yeah, and then the aggregate of the nucleosome will be built of the loop of the chromatin fiber here, yeah, and then the chromatin fiber pack also tightly join together and make the condensed section of the chromosome. And if we see in the chromosome, actually this part is one small part of the chromosome that like this. Yeah, this chromosome is in the form in the metaphase. Yeah, in the metaphase, we can see the chromosome like X letter. Yeah. Okay, and then we continue about the gene. What is the gene? Yeah, some students also confuse what is the gene and what is the DNA. DNA is the molecule, yeah, macromolecule that contain or that consists the unit named as nucleotide. And but what is the gene? The gene is the DNA fragment containing code or instruction for protein synthesis. So here you can see schematic diagram. Sorry. The gene A, yeah, will express, yeah, with the gene expression to build or to produce the protein A, and the gene B also express, yeah, with 2 phase of the gene expression, yeah. Later I will explain about this to become the protein B. So the gene C also will express to produce the protein C also. Okay, this is illustration how the gene express and then produce the protein. So now we continue about the human genome. So if you have already got information or understand about the DNA and the gene, and then how or what is the human genome? How many gene, how many DNA that we have as a human? Yeah. So here in the history, how we can now today the sequence of the human genome. So from the history, in the year 1985, 85, a project, with the goal of determining the complete nucleotide sequence of human genome was first formally proposed. There were some scientists, initiate to complete the nucleotide sequence of human genome. But in 5 years later, in 1990, the Human Genome Project, or HGP, was officially initiated in the United States under the direction of the National Institute of Health and the U.S. Department of Energy, with proposed timeline of 15 years, with the budget about the $3 billion US dollar, plan for the completing of the human genome sequence. This is the Human Genome Project consists or collaboration from the more than 10 countries in the world. I don't know, maybe as in Asia, Singapore include in this project. Okay, and in the year 2001, Venter et al. Published a nearly complete sequence of the chromatic portion of the human genome, and the sequence was performed by whole genome random shotgun method with subsequent assembly of the sequence segment. So I use also the publication from this author, Venter et al., in my lecture, because so far as I know, this publication is complete, explain about the process to complete, to sequence the complete human genome. So later I would like to show you the result of this publication, the result of this research. And I saw in this publication, there are so many scientists, maybe 100 scientists work together to work this task to complete the human sequence. So, and then in the year 2003, an accurate and complete human genome sequence was finished 2 years ahead of the schedule, earlier, and then at the cost less than the original estimated budget. So with the development of the machine, sequencing machine, this project finished earlier, 2 years earlier, and then also cost less than the proposed budget. So here you can access the human genome with this website. Okay. So now I will explain about the human genome, what the size, and then what is this inside of the genome. So this is the summarized information about the human genome. So human genome consists of 23 pairs of the chromosome, and 20 pairs chromosome consists or compose or consists divided into 22 pairs of the autosomal chromosome, and also 1 pair of the sex chromosome. In woman, a woman has chromosome sex XX, and then in man, has XY chromosome. And here you can see the picture of the human chromosome, from this source, human chromosome, Human Genome Resource at NCBI, National Center for Bioinformatics, Biotechnology Information. This is the website that's developed by NIH, National Institute of Health, like U.S. Ministry of Health, U.S. Ministry of Health. So don't forget also human genome, consists, composed also by copies of mitochondrial chromosome, or MT. So you can see here, I made the screenshot from this website. So if you go to this website, you can click this chromosome and you can see the mapping of the chromosome 1 until the chromosome 22. Yeah, chromosome, human chromosome was arranged, yeah, with the then for the classification or categorized and classified, yeah, during or through the size, yeah, from the big chromosome to the small, yeah, the biggest chromosome to the smallest chromosome. Here, yeah, you can see the number of the chromosome. And as I mentioned before, if you want to know more about the gene and the mapping of the gene of the nucleotide in each chromosome, you can click, yeah, this number of the chromosome, and you can see all about the sequence and the gene in the chromosome particularly, yeah. So, yeah, the information from this website so complicated, yeah. I can not also so good understand about the mapping and the gene or how the arrangement of this mapping, yeah. But maybe you can also access the information from this website. So here you can see the chromosome X, yeah, the bigger chromosome than the Y chromosome here, yeah. And as I told before, the woman has 2 chromosomes from this X chromosome, but a man has X and Y chromosome, and then another chromosome is a mitochondrial chromosome or mitochondrial DNA. Here we cannot see the picture about this because the mitochondrial chromosome is so small. Yeah, later I would explain to you about the mitochondrial chromosome or the DNA. So now the characteristic of chromosome in human genome. I got, yeah, this information also from the website from the NCBI. Here I think the updated information about the human genome. That is interested because as I read some textbook, yeah, there are so many different information about this amount of the gene, yeah, and also amount of the nucleotide. One book stated that the human gene in the gene on human genome has 20,000 gene. Another book stated 25,000 gene, and another book also stated until 30,000 gene. But I think if you access this website, you can also find the updated the amount of the gene or the characteristics of human genome. So here you can see the data or the characteristic of each chromosome, yeah, chromosome 1 until chromosome 20, and then chromosome X and the chromosome Y, and also the chromosome from the mitochondrial. Here it is the number, different sequence. Yeah, you can click this to get the many information about the chromosome 1. But another category, another numbering of this data, yeah, another data that collected by so many scientists can also with another code, yeah, like this, yeah, International Nucleotide Sequence Database Collaboration, yeah, also make the data with another platform, I mean, yeah. So actually until now there are 100, this is megabase, yeah, 183 million, million base, million base that is unknown, unknown. So here from this data we can see, now, until now, the scientists cannot understand or cannot identify the unknown sequence or unknown nucleotide. What is this, without any information, I mean. So here you can see the biggest chromosome, chromosome number 1 with the size 248 megabytes. It's mean that 248 million base pair. With the GC content, about 42.3%. How the important we can see, we can know about the GC, because from the GC content, we can determine how the DNA can be separated. The double helix DNA can be separated, because between G and C there are 3 hydrogen bound, as I mentioned before. So, and then here the data or the information about the protein, in the chromosome 1, a code, for a gene that code consists of the gene that code until 11,000, more than 11,000 the protein. And then in the chromosome 1, there are also 70 gene for the RNA, ribosomal RNA, and then 90 kind of the tRNA, the gene for the tRNA. So there is also other RNA like microRNA. There are so many kind of the RNA. Maybe in another lecture I explain to you about this RNA. So in this chromosome 1, the biggest chromosome, has until 4,000, more than 4,000 gene to encode the other RNA like cRNA, microRNA. There is also some kind of the RNA. So the interesting information is about the gene. The gene in the chromosome 1, the biggest chromosome, is until 5,000, 5,000 gene, and there is also the pseudogene. Pseudogene is the gene, but there are some mutation, maybe not completely gene, so in the pseudogene cannot express. And the chromosome 1, there are so many pseudogene. And then you can see here another information, in the chromosome, another chromosome, chromosome 2 until chromosome 20. So here, and the mitochondrial DNA here, the size is 2,000 to 20,000, about 20,000. But later I would like explain exactly how many nucleotide contained in the mitochondrial DNA, a chromosome, I mean. So here you can see the data. Once again, how many gene that belong to the chromosome, each chromosome in human genome, I mean. So, and then there are also unknown nucleotide, until 183 million DNA for 7,332 protein, and then and the gene also is from this unknown nucleotide sequence is until 7,000, and the pseudogene is 2,000, more than 2,000. So the totally size of the human genome is 3.2 billion base pair, yeah, with the protein, with the protein 123, more than 123 kind of the protein. And about the gene, yeah, I have already mentioned there are so many different information about the how many gene in the human genome, but here in updated data from this website, there are 61,000 gene, yeah, with the 18,000, more than 80,000 pseudo gene. So I mean, if we we see if we see this gene with the pseudo gene, it's mean that the gene that code in the protein is minus 61,000 minus 18,000. It is about 42 gene, yeah, encode the protein here. So okay, now I would like to inform you about the mitochondrial DNA or the chromosome. Excuse me, I would like to, sorry. So here is the diagram schematic from the human mitochondrial DNA. So here, yeah, the human mitochondrial DNA chromosome, yeah, contains, yeah, 2 genes for the RNA, yeah, the special RNA to build the ribosome. Yeah, ribosome is the suborganelle that play a role for the protein synthesis. So in the mitochondrial chromosome, yeah, contains two genes for the RNA. Yeah, there are 2 kinds, 16S RNA and 12S RNA. This RNA built the ribosome that play a role in the protein synthesis inside of the mitochondria. And then this mitochondrial DNA consists also 22 genes for the tRNA. The tRNA play a role for the synthesis, protein synthesis, yeah, that they can bring the amino acid, yeah, to the ribosome for the protein production. And then the important gene also contain in chromosomal mitochondrial DNA or chromosome is certain protein coding gene. Here you can see also the total length the genome of the mitochondrial chromosome is 16,469 base pair with the certain protein coding gene. Here you can see the NADH dehydrogenase subunit. There are so many kind of this enzyme, yeah, and then also there are 2 kind of the subunit for the ATP synthesis. There are also 3 isoform or T kind of the enzyme, cytochrome oxidase. Yeah. So the enzyme here that produced in the chromosome, the mitochondrial chromosome, through the protein synthesis inside in this chromosome, play a role for the oxidative phosphorylation that help, yeah, that carry out in the mitochondria, yeah, to produce the ATP. Yeah, the ATP is important molecule for our physiology even, yeah, in all of the organ in our body. So this is some information about the mitochondrial DNA. Yeah, mitochondrial DNA is susceptible to mutation, the high rate from the mutation. So until 10 times susceptible to mutation in comparison with the nuclear DNA. why the mitochondrial DNA is susceptible until 10 times for the mutation, because there is no DNA repair mechanism in mitochondrial DNA or chromosome. And also in mitochondrial DNA does not have protective nucleoprotein like in the nuclear DNA. You know, in my last slide, yeah, the DNA wrap the histone protein, yeah. So it make the DNA is stable, yeah. But in mitochondrial DNA there is no histone protein. Also, mitochondria contains the free radical substance. This free radical substance, like H2HO2, hydrogen peroxide, can damage the DNA sequence. Therefore, the mitochondrial DNA is susceptible for the mutation until 10 times. Here you can see the comparison, the characteristic between nuclear DNA and mitochondrial DNA. So in the size, you can see the nuclear DNA approximately 3.2 billion base pair. Yeah, I've already explained to you before in the last slide. And then the mitochondrial DNA, human mitochondrial DNA is more than 16,000 base pair. And the amount of the copy in the cell, yeah, in nuclear DNA, there are only 2 copies that come from the father and come from the mother, yeah. And then, but in mitochondrial DNA with the circular form, yeah, it could be the amount of more than 1,000. I have already read in some publication and the cell, the muscle cell, yeah, in our striated muscle cell contain more than 1,000 mitochondria. This high amount of it, the ATP production, you know, in the muscle contraction, yeah, need so many ATP molecule. Therefore, the mitochondria in muscle cell also so high. Yeah. And then the structure of the nuclear DNA is linear, packed into the chromosome. I've already explained to you how the nuclear DNA packed, yeah, in the chromosome inside in the nucleus. And then mitochondrial DNA in the form of the circular, yeah. And then the hereditary pathway from the nuclear DNA is maternal and paternal. I mean, the nuclear DNA can originally come from the father and come from the mother. But in mitochondrial DNA only come from the mother or paternal heredity, because you know the sperm, yeah, cannot enter the whole sperm, yeah, the whole body of the sperm cannot enter the ovum outside in the fertilization event. So the mitochondrial in chromosome located in the tail, yeah, in the neck of the tail, yeah. And then now the fertilization, in the fertilization, only the head of the sperm can enter the oocyte. It means that the mitochondrial sperm cannot combine, yeah, in the maternal DNA, yeah, to make a zygote, yeah, in the zygote. And here about the mutation rate, yeah, the nuclear DNA has a low mutation rate, but as I explained before, yeah, the mitochondrial DNA has a high rate in the mutation. So now I would like to proceed to the next information, how the gene organization, yeah, how is the organized gene in the chromosome. Here, as an example, here you can see the human chromosome 22, yeah, 22. This is the smallest chromosome, yeah. Here you can see the chromosome 22 has amount about 48 million nucleotide. Yeah, the small chromosome, but yeah, has a million nucleotide. And then 10 chromosome of this chromosome number 22, yeah, contain about 40 gene, and then one chromosome contain 4 gene. I think I have already explained to you with the last table, yeah, last table. So, and then here, yeah, you can see 1% of the chromosome contain only 4 gene, and then here you can see the part or the region of the one gene. Yeah, one gene, yeah, is about 3.4 10 magnification 4 in chromosome 22, yeah, composed by two part or two region, two mainly region. Yeah, it's been that regulatory DNA sequence here, yeah, and then the structural region, yeah. The regulatory DNA sequence is part or the region of the gene that facilitate the gene expression. Yeah, here in this region will be bind by transcription factor, yeah, to induce the transcription process in the gene expression. Here, another part, another main region of the gene, named or called as the structural gene region, composed of two kinds of the fragment, exon and intron. And then you can see in the gene expression, yeah, with if we see the process of the transcription, yeah, the first step in the gene expression, you can see the exon and exon, yeah, will be packed together, but the intron will be spliced, yeah, will be cut it, yeah, in the splicing process in the transcription. So after the transcription process, I will explain to you later. The mRNA, the result of the transcription process, will be translated in the ribosome to produce the protein, and then the protein will be undergone post-translation process like folding, and then make a folded protein that functionally, yeah, that functionally for the physiologic process in the cell or in the organ, yeah. So now I would like to explain to you about from the DNA, the protein here. You can see the DNA can make the same sequence like itself, yeah, with the DNA synthesis or replication, yeah. The replication DNA happen at S phase in the cell cycle or synthesis phase in the cell cycle. But for the gene expression, yeah, the cell must do the gene expression, yeah, to serve the protein, yeah, that can function in so many physiological process in our cell, yeah. So the DNA will express with two steps, yeah, with the transcription process, the first step for the RNA synthesis, and then the outcome for the transcription process is the mRNA, yeah, molecule. And then mRNA molecule will be translated in the ribosome to produce the amino protein synthesis that composed by amino acid. Yeah. Okay, and how the gene expression happen? The first step is the transcription. Okay. Here you can see that there are so many protein, yeah, work together to induce the transcription process. I have already read, yeah, in some textbook stated that the 100 protein play a role or work together to induce the transcription. You can imagine 100 protein can work together or must work together to initiate the transcription. So here you can see illustration about the so many kind of the protein or transcription factor bind to the region promoter or gene regulatory segment of the gene, and then here you can see the many proteins work together to induce the transcription, and the result of this transcription is messenger RNA. This transcription process happen occur in the nucleus, and then the mRNA as a product of the transcription process will be delivered with the special transport system to the cytoplasm. The destination of the mRNA is a ribosome. In the ribosome, the mRNA will be translated. You can see here the illustration of the translation process, the second step of the gene expression. So here you can see the mRNA will be read, in the ribosome. The reading process of the mRNA actually use the codon, three nucleotide or triplet nucleotide that code for the specific amino acid. The amino acid will be brought by tRNA. tRNA bring the amino acid here, and then the RNA will enter to the ribosome, and in the ribosome, and it happen the growing polypeptide to make a protein sequence or amino acid sequence, completely amino acid sequence to build the a protein. So this is the illustrated how the translation process happened in the ribosome. Okay, now another information, another data about human genome or human gene. So I would like to inform you about the publication that published or that delivered the result of the research, again from this author, that who published the result. I have already told there are so many scientists work together to sequence the human genome, and the result of this research published in the Science 2001. This publication is reversed also from many textbooks. I think the information for this from this publication also valid. I mean, so okay. Here you can see there are so many protein, kind of the protein, produced from human genome. Here you can see the most protein that produced by gene expression in our genome, in human genome, is for this transcription factor here, nucleic acid binding. It's mean that transcription factor, and also for the signal transduction. So here you can see they are categorized of the protein that coded by our genome. Here you can see for the structural protein of the muscle, there are 296 kind of the protein for the structure of the muscle cell. So here for proto-oncogene, there are so many also, 2%, 2.9%. There are 902 kind of the proto-oncogene. So here, this is interested data about the percentage of the protein that produce in our genome, in human genome through the gene expression. So there is also, the most protein is from the signal transduction here, and then the enzyme, there are many categorized, many kind of the enzyme, transferase, synthetase, oxidoreductase, liase, this with the percentage of the protein in all of the protein that produced in our body, in our cells. So this scientists, categorize or make a research with two kind of the system. The first system is with the gene ontology, system, and then the scientists also use the spatial database, smart database with the Celera Panther, and then make this chart, with two different categories. Geo-categorized here in the outer side circle, and then the Panther categorized. It's not so important, but the result of this research, the scientists, or we can see there are so many protein, and then also the percentage, of the protein from each kind of the protein, like this, Okay, and then now we proceed about the human genetic variability. So actually in year 2003, the Human Genome Project, already also completed, identified of more than 3 million human genetic variation called a single nucleotide polymorphism. And this single nucleotide polymorphism, or SNP, caused by mutation. So this mutation can make the alteration on qualitative and quantitative of the gene expression product or the protein. So with these changes, or this with the alteration in the protein, qualitative as well as quantitative, it can be make the variation in human phenotype. So like we are different from each other. It's mean that there's human genetic variability between us. So human phenotype variation caused also by epigenetic mechanism. What is the epigenetic mechanism? Actually, epigenetic mechanism is gene, but the expression of this gene is different. It's changed with the cause, not from the mutation, but from another cause, another mechanism. So epigenetic, it's mean that epi is above or out of the genetic. If we learn about the genetic, we learn about the gene, genetic, gene, DNA, and also mutation. But in epigenetic mechanism and epigenetic field, we learn about the genetic, but the expression of the gene is not caused. It can be changes, but it could not by the mutation, but caused by another reason, such as DNA methylation, histone modification, and then non-coding RNA like microRNA or cRNA. So this epigenetic mechanism, these three categories of the epigenetic code or epigenetic mechanism can be influenced by nutrition. So I would like to show you the schematic diagram. What is the epigenetic mechanism? Epigenetic mechanism are affected by this factor and processes such as development, in utero in childhood, during the childhood, and then the environment chemical. The methylation maybe come from the chemical that present in our environment. And also the drug and pharmaceutical, yeah, can also as a source of the methylation or some substance that can modify the histone. Also aging, yeah, aging also is can affect the epigenetic mechanism, and the diet, nutrition, yeah. Now we learn in the global academic program on nutrigenetic and nutrigenomic, yeah, we focus learn how the diet or nutrition or nutrient can affect our genome, yeah. So here you can see the DNA methylation here. The methylation bind to the special base in the DNA, the base nitrogen base, and DNA methylation can repress, yeah, the gene expression, yeah. So here you can see the DNA sequence is not mutated, not altered, but the expression of the gene is altered or is changes because the DNA, the methylation, yeah, bind to the region, a promoter region, gene regulatory protein, yeah. So the transcription factor cannot bind to the promoter region of the gene. So yeah, and then the histone modification, yeah, you know, the histone modification can also repress or activate the gene expression. It's depend on which substance can bind in the tail of histone. Yeah, the modification of histone will be conducted by acetylation, methylation, yeah, phosphorylation also, and then the substance can repress or can influence also the gene expression. So the health endpoint of the epigenetic mechanism, yeah, is a cancer, autoimmune disease, mental disorders, or diabetes. Yeah. So I think here, yeah, now we are in the nutrigenetic and nutrigenomic field. Yeah, we learn more about this nutrigenetic and nutrigenomic from another lecture for 2 days, tomorrow, yeah. Nutrigenetic is a study about the effect of the genetic variation on dietary response. Yeah, what is the difference between nutrigenetic and nutrigenomic? Yeah, so here in nutrigenetic, we learn about the effect of the genetic variation on dietary response, but nutrigenomic is science focused on the role of the nutrients and bioactive compound in the gene expression. So, okay, I would like to show you, yeah, the video that I got from YouTube. Yeah, I think with this YouTube you can understand better, yeah, better about the human genome. Yeah. Okay, I would like to play this YouTube about the introduction of human genome. Yeah, for the refreshing also. Okay. Before I continue with the YouTube video, is there any question from the participant? No? If there is no question, I would like to play the video about the introduction of the human genome. Yeah. Okay. Wait, please wait a moment.
Humans around the world have much in common, but...
Can't you hear?
Yes, Prof.
Yes, okay, thank you for the confirmation.
Also enormous diversity. Some of the differences between each of us come from our environments and life experiences, but our DNA plays an important role in determining our appearance, our traits, and our health. There are thousands of genes in the human genome. Sequence changes in individual genes can determine if we have freckles, can digest lactose, have wet or dry earwax, are red-green colorblind, or are likely to have blue eyes, or think broccoli tastes bitter. Individual genes can also determine if we will develop sickle cell anemia, cystic fibrosis, or Huntington's disease. Multiple genes act together with our environment to determine our hair and skin color, our height, our weight, our blood pressure, and our risk of developing type 2 diabetes, depression, cancer, some autoimmune disorders, and many other conditions. In spite of all these potential differences, humans are 99.9% genetically identical. How is it possible that we are all so similar and yet so different? Let's zoom in to the smallest genetic unit, a single nucleotide of deoxyribonucleic acid, or DNA. A DNA nucleotide is composed of sugar and phosphate groups and one of 4 nitrogenous bases: adenine, thymine, guanine, and cytosine, annotated in shorthand as A, T, G, and C. The sugar and phosphate groups form the DNA structural backbone, allowing nucleotides to concatenate into a long single strand of DNA, while the bases determine DNA sequence. The chemical properties of DNA allow bonds to form between the bases in order to create a double strand, with 2 hydrogen bonds pairing A and T, and 3 pairing C and G. Though different types of human cells can be very different in appearance and function, they contain the same genome, which consists of about 3 gigabases, or 3 billion base pairs of DNA. All the DNA in the cell would be about 2 meters in length if it were stretched out, and must be condensed down to fit into cells as small as 10 micrometers across. The DNA is first coiled into its canonical helix structure, and then wrapped around histone proteins to form a DNA protein structure called a nucleosome. These nucleosomes can be further wound and coiled together to create a compact structure that fits into the nucleus. During cell division, the DNA is organized into tightly wound chromosomes, 46 in total, with 23 coming from each parent. These chromosomes can be easily and accurately separated during cell division, guaranteeing that each new cell contains an exact copy of DNA. Outside of cell division, the DNA is decondensed in the nucleus, allowing greater accessibility. The transcriptional machinery regulates expression of the approximately 20,000 genes, which, although they correspond to less than 2% of all genomic DNA, encode all the proteins necessary to build and run a human cell. So to return to the original question, how is it possible for all of human diversity to exist when we are 99.9% genetically similar? It is important to remember that the 0.1% of DNA that varies, on average, between each of us actually corresponds to about 3 million differences across the genome, with 20,000 of them, on average, falling into protein-coding genes. Although that equals approximately 1 difference per gene, in reality, these differences are not evenly distributed across the coding regions. Differences in DNA sequence are called variants, and those affecting a single position are called single nucleotide variants, or SNVs. Common SNVs that occur in more than 1% of a population are called single nucleotide polymorphisms, or SNPs. SNPs, along with larger scale sequence changes like deletions, duplications, and rearrangements, create all the richness of human genetic diversity at the population level. This raises many questions on an individual level as well. How does DNA determine our traits? How can we understand what a gene does and how variants in that gene might affect our lives? What can our DNA tell us about our risk and our loved ones' risk of disease? How can information from our genomes improve our medical care? Understanding genetics allows us to apply the concepts of heritability and genetic variation to questions of human health and disease in our world today.
Okay. Okay. So I continue with the take-home message. Yeah? Okay. So I hope that you can better understand with this video about the genome. Yeah. Also the human genetic variability that determine our different phenotype, yeah, the different phenotype between human, yeah, also for the disease, yeah, responsible also for the disease. So here I would like to continue my presentation, the last slides about the take-home message from my lecture today's, yeah. The human genome contains more than 3 billion nucleotides called as DNA or deoxyribonucleic acid, and then more than 40,000 genes. Yeah. I would like to make stress in this point of information about the how many genes that we have. Because yeah, in different textbook you can find different number or different amount of the gene, but from the website that I got for lecture today, yeah, the gene, yeah, protein coded gene, yeah, present more than 40,000, yeah, because the knowledge, the science is developed so far, yeah, with the finding of the sequencing machine. So, and then the scientists also explore the protein, yeah, the protein and the function of the protein. The important thing is the function of the protein, and then this many information, it can make that different data or different information, yeah, that we can get, yeah, also in the future, I think. And then this human genome, yeah, this gene or this DNA, more than 3 billion nucleotides packed into the only 46 nuclear chromosome, yeah. I've already show you, yeah, there are so many chromosomes, 20 pairs, yeah, chromosome, autosomal chromosome, and 2 chromosome sex, and also about more than 1,000 mitochondrial chromosome. It can be less than 1,000. Yeah? It's depend on the cell, yeah. So more than 1,000 mitochondrial chromosome located or present in the muscle striated cell, yeah, in our body, yeah. So, and then the second message from my lecture again is expressed, yeah, for the protein synthesis, okay. And then human genetic variability, yeah, caused by mutation and epigenetic mechanism can cause the alteration on the protein function, may influence the human health, yeah. And then the epigenetic mechanism can be influenced by nutrients that we would like learn in this module or in this global academic program. So this is the slide take-home message, and thank you for your attendance and also your attention in my lecture today. Okay. Now we open for discussion. Is there any question? Before I finish my lecture today or our discussion today about the human genome, I would like to inform you detail about our program, yeah, genetic global academic program. So now Is there any question? I saw here.
Yes,
Sorry.
Prof.
Yeah,
There
yeah.
are two questions.
Okay. Chat room. Okay. Would you inform us keyword to see the presentation through the canal YouTube? Okay. And then the question to Prof, is epigenetic damage permanent? So to cure the disease, cancer, autoimmune, etc., is enough just to repair the histone modification or DNA methylation from the Nurfaiza. Okay. Epigenetic damage permanent. Epigenetic is not give negative impact in our physiologic process in our body. Yeah, epigenetic mechanism is normal process in the gene expression to make the balance about the protein synthesis or to produce the suitable amount of the protein. So the epigenetic can be changes, as I mentioned before, the epigenetic mechanism can be changes, during the childhood, during the fetus life, and also we can also acquire genetic code. We can have also acquired genetic material or genetic code, like drug, and pharmaceutical agent. It can be the mechanism for the gene expression alter, changes. So I mean, epigenetic damage is not so exactly true about the damage. With this context, the damage, because the epigenetic is normal. You know, the zygote, the one cell of result of the fertilization. All of our DNA is methylated. I have already read the 100% of genome in the zygote is methylated, but during the zygote development to make the fetus, it can be the methylation percentage will be decreased and decreased, and then this is continue with the organogenesis. It's mean that it can be this the epigenetics not permanent. Okay. So and then to cure the disease, cancer, autoimmune, is enough just repair the histone modification or the methylation. Okay, about the treatment, as far as I know, the one disease that can cure or that can treat it by a substance that can reduce the DNA methylation is the myelodysplastic. This that can continue to the leukemia. There are some publication that publish special substance like 5-azacitidine. 5-azacitidine can reduce the methylation percentage of the methylation in the gene, especially in leukocyte. So this substance, the 5-azacitidine, has already approved by FDA, Food and Drug Agency like Badan POM in Indonesia, to cure or to treat for the myelodysplastic patient. It means some substance, another substance can cure the disease like the myelodysplastic ini. So about the histone modification, I don't know exactly. Honestly, I don't know about the how or so far how the histone modification so far use the drug or the substance can reduce or can modify the modification of histone for treatment to the disease. Yeah. I don't know about the histone modification, but DNA methylation, I think, yeah, I have already read that 5-azacitidine can reduce the percentage of DNA methylation that treated for the leukemia, not leukemia, myelodysplastic patient. So, yeah. Maybe I should read some publication more for the histone modification, yeah, for the cure, yeah, cure the disease in human disease, I mean. So, and then here another question, siapa tadi? Nurfaiza. Okay, Nurfaiza. Are you, it is enough from my answer?
Yes, it is enough, Prof. Thank you.
Okay, I want to know how come from you? Where do you come from?
From Unpad, Prof.
Unpad. Oh, nice. Okay. Salam ya buat orang Unpad. Okay. And then, okay, Duyanti. Yeah, the question, 2 questions from Duyanti. This is my student. Okay, dear Prof, I would like to ask 2 questions. What is the importance of GC content in our genome as well as in the molecular techniques in like PCR or sequencing? So I would like to answer the first question. Yeah, the first question. So GC content, yeah, like as we see in my slide, yeah, the GC content is also one examination point, yeah, that would also important that we should know, yeah. Also, GC content in our genome make the DNA, yeah, more tightly, more in the double helix. I mean, because between GC there are 3 hydrogen bound, yeah, that can, yeah, difficult to separate, yeah, the single strand from another strand. So I mean, for the replication, yeah, DNA replication, the double-stranded DNA, yeah, must be separated, yeah. If the GC content more or higher, yeah, it can be that the denaturation of the double-stranded DNA into the single-stranded DNA is difficult, yeah. And as we know in the PCR, yeah, PCR is actually a replication process, yeah, DNA replication process in the lab, in the in vitro. Yeah, the GC content, yeah, should be also make the thing that should be pay attention, because with the GC the higher GC content, it can be the denaturation process from the double-stranded DNA will be difficult, yeah, and we should use the higher temperature, yeah, to break the 3 hydrogen bond between G and C bound, yeah, in double-stranded DNA. So it can be the GC content, higher GC content in our DNA, yeah, fragment DNA for PCR method. It can make the annealing temperature is higher, yeah, is higher. And then we should not use the high annealing temperature for the PCR or sequencing method. And then the second, yeah, Duyanti, can I answer your question enough?
Yeah, Prof, very clear. Thank you, Prof.
And then the second question, as mitochondrial DNA is very susceptible to mutation and can be inherited from mother to the children. What is your opinion about the mutation in maternal mitochondrial DNA affecting the child health? Yeah, of course, yeah, the mutation in the mitochondrial DNA can affect to the child health. Yeah, if the mother has disease, yeah, that caused by mutation in mitochondrial DNA, it can be delivered, yeah, to the child, yeah, from to the boy or from also to to the boys, yeah, to girl or to the boys, and the daughter that affected by this mutation, it can be also delivered this mutation, this disease to the next generation. But the a boy, yeah, a son from this suffered mother, it cannot deliver more this disease to the next generation. It can make that maternal heredity. And about the severity, it's also different. Yeah, it's depend on the how many affected mitochondria can deliver to the offspring. Yeah, it can be the mother is not suffer from this disease, but the next generation, the daughter or the sons, can be that more suffered. Yeah, from the mother. Yeah, there are special mechanism, bottleneck mechanism in mitochondrial inherited. Yeah, so maybe I will explain about this in another lecture. Okay, and then Gurit, yeah, Yanti, is enough my answer? Okay, and then Gurit. Therefore, I just wonder, can nutrient can change nitrogen base, which is defect? It can make protein is not function normally. So as I explained before, yeah, the nutrient can make epigenetic mechanism. Yeah, not cannot. Yeah, the nutrient cannot make the mutation. Yeah, it can be. Yeah, it can be, but yeah, like UV light. Yeah, UV light can make the thymine. Yeah, thymine change to the uracil, and then this make mutation. Yeah, but the most mechanism that caused by nutrient is epigenetic, because the substance from our nutrient can influence, yeah, the methylation of DNA, modify our histone, yeah, phosphate group, yeah, methyl group, acetyl group, yeah, that may be contained in our nutrient. It can be bind to our DNA or our histone, yeah. Okay. Jadi and then it can make protein is not functionally normally. Yeah, if the mechanism, epigenetic mechanism occur caused by the nutrient, it can be the protein, yeah, the production is not enough. Yeah, so the gene expression that caused by epigenetic will influence the quantitative of the protein, not the qualitative. Yeah, the quantitative. Yeah, whether high protein production or less protein production. Yeah, not the structure of the protein. Yeah, protein that produced by epigenetic mechanism also cannot function. Yeah, because the number or the amount of the protein is not enough. Yeah, for example, the enzyme, yeah, should with enough amount to reduce or to catabolize the substrate. If the amount of the enzyme is low, yeah, a little bit, yeah, it cannot destroy the substrate. Yeah, it can be disease. Yeah, it can make the disease. Yeah. Okay, and then another questions about is epigenetic correlated with the nucleic DNA or mitochondrial DNA or both? Epigenetic that correlated is in the nuclear DNA, yeah, nuclear DNA. Or whether can affect also in mitochondrial, also especially in DNA methylation. Yeah, DNA methylation can affect in into the nucleic acid, but in mitochondrial DNA, until now I haven't read about the methylation DNA in mitochondrial DNA. Yeah, I don't know. Maybe I haven't read so many publication about DNA methylation in DNA, mitochondrial DNA. But in nuclear DNA, of course, yeah, our gene can be affected by methylation. Yeah, one of the three of epigenetic mechanism.
Thank you, Prof. Clear.
Okay. Another question, maybe with the raised hand. I saw one person raise hand. Okay, no question. So if there is no question, yeah, now 11, yeah, 40 minutes after 11. Okay, I would like to inform you about our program. So let me inform you about our program now. Okay, so yeah, is there any question? If there is no question, I continue with this announcement or our information, the information from our program. So no, yeah, okay. There is a prize hand from the Gede Wirata.
Gede Wirata.
Okay, yeah, please, Gede. Where do you come from? Can you
Thank you
inform
for the
me?
invitation for this important meeting, Prof.
Yeah.
Okay,
Okay.
Prof, I would like to ask you about the result of DNA test. Mostly people in Bali, because I live in Bali, and mostly people in Bali confused about their choose their option after getting DNA test for their
what kind of DNA test? DNA test?
Yeah, they confuse to whom doctor they would like to present their results and for consultation, Prof. Like that, Prof.
What's the aim of this DNA test? To know what's the aim of this?
Because as for epigenetic progress nowadays, they maybe would like to know about the important side of epigenetic knowledge to their disease, maybe like cancer or chronic illness. So they are confused to whom doctor they would like to present their result or just for consultation, like that. Because as we know, we have lack of genetics practitioners in Bali.
So as far as I know, people want to make or want to evaluate the DNA DNA test, to know the susceptibility this person to the disease, for example, for the cancer, for example, Yeah, in many developed countries, yeah, this DNA test to evaluate the susceptibility or the risk factor evaluation to disease like heart disease, yeah, for example, in Netherlands. Yeah, the people can get the result from the human genetic to evaluate or to measure the score, how high this person to get this disease like heart disease, for example. Nowadays, there are so many lab also performs this test to evaluate the risk of the disease. But today, nowadays I have all the information. The leukemia, the treatment of the leukemia, it's depend also from the mutation that may happen in the some regions in the chromosome 9, actually. I don't know. Yeah, exactly. 9, yeah. There are special mutation from the BCL-ABR. I don't know the fragment. I forget the fragment. Yeah. If there is a mutation, in this leukemia patient, the medical doctor, the clinician, cannot give more the first therapy, first agent, to cure the leukemia. If there is mutation T151 to I don't know.
Kamu Irfan, lo ke FK aja nggak apa-apa.
In this gene, BCL-ABR gene, I don't know what's happened. I forget that. If there is a mutation, T2 in the region in the amino acid 151 to another amino acid isolation, so the patient should be given another pharmaceutical agent, ponatinib, kalau nggak salah. This is second line for the leukemia treatment. So with the DNA test, like to evaluate the mutation in the gene and spatial target, we can cure the patient with special drug. So the benefit that this DNA test is for the identify or to determine the high risk risk factor for the disease and also for the cure of the disease, to treat the disease. Itu jadi, but the lab should be facilitate with this DNA test or the gene test. So gede, jadi can my answer is enough for you?
Yes, that's enough, Prof. So the conclusion, we know the DNA test result only for determine or identify the high risk or lower that is, Prof. Thank you, Prof.
Yeah, it can be. Also, we can also analyze the DNA for the identification of the person, like in forensic medicine, to identify the cadaver that cannot recognize more because the damage from the body. It can be identified by DNA test, the fingerprint test. This test evaluate the repetitive sequence of the genome, in the human genome. Actually, the repetitive sequence in our genome is so high. 50% of our genome, of 50% of our human genome, contain the repetitive sequence. So tandem repeat sequence, the sequence that repeated how many times? So 50%, our genome contain of the repetitive sequence. So, and then this repetitive sequence will be evaluated in forensic medicine to identify the persons. Yeah. Okay. So, yeah, Gede, is enough.
Thank you, Prof.
Yeah, thank you. You're welcome. Okay, and then I would like to inform you about this, about our program. So, as I mentioned before, that our program is Global Academic, or GAP, on nutrigenomic and nutrigenetic, or nutrigenetic and nutrigenomic. This is the lecture series, in this module that has 4 credit point for the participant that will get the credit, or credit earning for this, for many purpose, such as for the education in master program, maybe, or in doctoral program. We provide this lecture, this program, on nutrigenomic and nutrigenetic. So here actually the whole syllabus, the whole lecture that we provide you if you want to join in this program. So with the yellow highlight is open sit-in like today about the human genome that was delivered from me today. And then here with the yellow highlight, and the next today, Professor Paul Hagarty. Don't miss this event. I interested also to join in this lecture because I will also learn about this from the Professor Paul Hagarty. So don't miss this event. And then another lecture that free, sit in, for the participant. All of participants by availability of nutrient will delivered by Dr. Umi Famida from SEAMEO RECFON. And also here again from the Professor Hagarty from Aberdeen University, about this theme or about the topic. That's also interested topic. And then about the epigenetic and gut health microbiome from the will delivered by Dr. Anauska from Aberdeen University at on I don't know when the day, 25th November 2021. So I would like to explain to you more. Actually, we provide so many lectures from this program, with 4 credit point with 4 learning objectives. So in first learning objectives is about the understanding concept, the human genome and the gene. So after my lecture, actually we'll continue with the another lecture, Pak Dwi Ari. I show Pak Dwi Ari also attend in this lecture, my lecture today, human genetic variability. And then another lecture from me again is about the principle and practice of genetic analysis technique, may be interested for you. But here the lecture is just for simple technique like PCR, qPCR, Q real-time PCR, methylated specific PCR to evaluate the methylation percentage of the DNA in our genome. Then RLV, restriction fragment length polymorphism, and also sequencing with Sanger method. But here also interested topic will be delivered from Dr. Anioska from the Aberdeen University, principle and practice of genetic analysis technique method, with the advanced method such array, epic array, to genetic and epigenetic analysis, and also a sequencing method for genetic and genetic analysis. You know that now there is a special technology for sequencing named as next generation sequencing. This is the method, the application that is sophisticated and also expensive, of course, yeah. But with this method, I think we can sequence. Yeah, the rate of the sequence is so enormous, yeah, and so fast also. So, and then here with the yellow highlight is open for sit-in, yeah. You can join this lecture free, yeah. And then another is the evaluation, yeah, for the student that want to get the credit point, yeah. There is the evaluation from us about the knowledge of the student, yeah, or the participant, not only the student, with the presentation and discussion and journal operation and evaluation. So, and then second learning objective, yeah, understand the principle of genome and nutrition interaction. Here, there are so many lectures about the nutrition, yeah, from the basic nutrition and nutrient requirement, dietary assessment, biomarker and nutrient status, yeah, from will delivered by Ibu Umi Fahmida from SEAMEO RECFON. And also here the highlight, the yellow with yellow highlight, yeah, this is free. Bioavailability nutrient also will delivered by Ibu Umi. And then also the next interested lectures is about nutrigenetic and nutrigenomic application of omic technology in optimization of human nutrition. It's also interested topic, yeah. But yeah, unfortunately, this lecture is not open free. This is for the student that register or enroll in this program. So please, if you're interested in this program, yeah, maybe join this program in next year, yeah, because we arrange this program every year, not not every semester or not every month. So, and then once again, the free lecture from Professor Hagarty here with the highlight, the yellow highlight. And then after lecture from the second learning objective will be also evaluated, yeah, with the presentation and discussion from from the student or a participant with journal appraisal. And then here for the third learning objective, understand the principle of microbiome nutrition and epigenetic interaction. Here there are so many lectures also. Here the first lecture in the third learning objective is introduction to bioinformatics and metagenomic studies, will be delivered by Pak Dwi Ari, this staff or lecturer from medical department, medical biology department, like me in Faculty of Medicine, Universitas Indonesia, and also delivered by Dr. Felix Mesah. This is alumni from our study program, who has a biotechnology company in Canada. So he has so many experience, yeah, in the bioinformatics and bioengineering. And until now in Canada, he has also a student, yeah, that is supervised, yeah, by him, yeah. Okay, and then another lecture, the human microbiome and metagenomic studies, will delivered by Dr. Alan Walker. He come from the Aberdeen University, Rowett Institute. So also interesting topic. And then here with the highlight yellow highlight, this Dr. Anouska will deliver a topic about the epigenetic and gut health microbiome. This is free chart for the participant. Yeah, and then the contribution of microbiome to health and disease also will delivered by Dr. Felix Mesah. From DNA Tracia Biotechnology Company in Canada. So, and then the evaluation will be performed until after the lecture in this third learning objective. And then in the fourth learning objective, there is no more lecture, but here is the journal appraisal for the student, yeah, that get the credit, yeah. Will the mode of the presentation, yeah, will evaluate the student, Yeah, and then there are specific topic for this session. Yeah, how the nutrigenomic and nutrigenetic play a role in the cancer. Yeah, and we give the publication journal to evaluate and to discuss together, and also for the cardiovascular disease, obesity, and metabolic syndrome and diabetes. So this program will end in 17 December 2021. So maybe you can see in our announcement that the date for this free lecture is so far, yeah, 26 October, 27, yeah, and then the next is 15 November. Yeah, maybe you can see here, but actually in between lecture by lecture, that free is there are so many lecture. Yeah. Okay, I think that's all for the information about our program. Thank you very much for your attention and your attending, of course. Okay. Okay. Rizky, sudah kita aja, kita tutup. If there is no more question.
I would like, before we close and end the session, I would like to express my gratitude to our speaker, Professor Dr. Asmarinah, for her delightful information and insightful presentation. And as we are now at the end of our session, I would like to take a group pictures with all participants. To all participants, please turn on your camera.
So there are 5 layers, yeah?
Yes. Okay, from the first page. 1, 2, 3. Okay.